home and work at it. You have Monday off, right? After barbecue and everything you do, just
Yep.
complete and bring it next Wednesday. Okay? Next Wednesday is our last session of the lab, and then the week after we have lecture exam, last lecture exam, and last lab practical. So in order to manage this effectively, I have to discuss the reproductive system of the male. So the reproductive system of the male consists of two distinct parts. One is the primary sex organ of the male, which is testis, and the other one is the accessory or secondary organ. And the secondary organs of the male reproductive organ include ducts of the reproductive organ and glands of the reproductive organ, okay? And then we have external genitalia that its function is to basically excrete the urine because urethra is there, and also it's the erectile organ in male. We're going to very briefly discuss it. I saw that they have only put for us one male pelvis, and then we're going to look at the histology of the testes, which is very, very important, and it's all lab practical. So after I discuss it, I'm going to show you what the organs are, and I am really, really summarizing it. Now, the duct of the reproductive organ of the male, I say it briefly and then I show it one by one. So include epididymis, ductus deferens, ejaculatory duct, prostatic urethra, membranous urethra, and spongy urethra. And then the gland of the male reproductive organ are three of them. One is prostate gland, seminal vesicle gland, and bulbourethral gland. So we just briefly outline their function, that's it. There is no histology of these things recorded. I talked to the coordinator, the cadaver is not on the lab practical, even though you see in that guide that she gave you, it says cadaver and cat, but it doesn't apply to you, okay? So just study the structure that we have around us, okay, and histology. So let's start with the testis. Did you know that the testis during embryonic development is inside the abdomen? And it actually, when the embryo develops, testis is inside of the abdominal pelvis area. But right when the baby is born, the fetus is born, the testis take a piece of peritoneum, which is a serous membrane of the abdomen, and then take a piece of skin with it, which we call it the scrotum, and exit out. Now the question is, why this evolutionary adaptation? Why testes must exit outside of the body after birth? In fact, children that are born with their testes inside the pelvis remaining and doesn't come out, they are infertile. Did you know that? Because spermatogenic cells, which are stem cells or precursor cells for spermatogonia, are very, very sensitive to physiological temperature of the body, which is 37 degrees. They don't survive and they die. So right before birth, testis, which contains a spermatogenic cell, exit from the body and remains outside of the body, and that keeps the testis 3 degrees cooler than physiological temperature. So 3 is not absolute number, okay? So -3, 3 degrees. When the spermatogenic cells become spermatozoa during the 4th step of differentiation, they can tolerate 37 degrees, but not a spermatogenic cell. Those are precursor for spermatogonia, which produce spermatozoa. Now, if you look at the structure of testis, it is divided into lobules by these tubules, narrow duct. they call seminiferous tubules. Seminiferous tubules. In fact, what you're going to look under the microscope is the histology of the seminiferous tubule, in which you detect a spermatogenic cell, primary spermatocyte, secondary spermatocyte, a spermatid, and a spermatozoa. So for now, let's look at this one. So each lobule of the testis is formed by this connective tissue, tunica albuginea, and then you can see each lobule contains 1 to 4 seminiferous tubule. So the testis of the male is the factory for synthesis of spermatozoa. That's one function. And its second function, it produces a very important hormone called testosterone. Testosterone is very important for spermatogenesis, and also it's very important for secondary male characteristics, such as development of massive muscle, beard, pubic hair, and etc. So second male characteristic. Now guys, there are 4 types of cells in this seminiferous tubule, and those cells, I'm going to show it here. The first one is epithelium of the seminiferous tubule, okay? The epithelium of the seminiferous tubule contains a spermatogenic cell, which are the cells that produce spermatozoa, but the spermatogenic cell must go through 4 stages of differentiation. The spermatogenic cell is first, 2, primary spermatocyte, 3, secondary spermatocyte, the spermatid, and finally the sperm. Now, sperm plural is a spermatozoa. We don't put the S and call them a sperm. So you have to write a spermatozoa or a sperm. Now, there is another type of cell. It's called sustentacular cell, also known as Sertoli cell. The sustentacular cell are supporting cell for the spermatogenic cell and all the differentiation stages of the spermatogenic cell. So they support. Basically, guys, you can think of a spermatogenic cell as a neuron, and these Sertoli cells are neuroglial cells. So, because actually they have very similar function to astrocyte, means that they create blood-testis barrier. Blood-testis barrier. The spermatogenic cells are so delicate and sensitive that sustentacular cells protect them from coming in contact with blood by forming tight junction, just like the astrocyte. Tight junction that prevents passages of unwanted, undesirable molecules in the blood into the testis. Now, sustentacular cells have several important functions. I'm going to mention 3 of them. One, supporting the spermatogenic cell and all their differentiation state via creating tight junction, okay? So that they don't become in contact with blood. So they create blood-testis barrier, that's 1. 2, they produce a protein known as testosterone binding protein. Testosterone binding protein, and this testosterone binding protein, its name describes its function. Who can tell me the function of testosterone binding protein? It really tells about its function. Go ahead. One of you. Anybody? Okay. Yes.
Binding testosterone.
Thank you. Yes, it binds testosterone. Why this is important, guys? Because testosterone secreted in low amount, and in order spermatogenesis to happen, this protein bind many of this testosterone and in a way concentrated in the testes for spermatogenesis. That's two. Now, the sustentacular cell or Sertoli cell or sustentocide also are responsible because the sperms are, cytoplasm is very big. If you look at the spermatid, you can see the nucleus is this much and there is a lot of cytoplasm. So those spermatozoa shed their cytoplasm, and who takes them up? The sustentacular cell. So they actually phagocytize the excess cytoplasm from the spermatid that shed. Okay? Now, the third type of T cell are called myoid cells. Myoid cells are located in the wall of the seminiferous tubule, and they are like smooth muscle. They basically create contraction that creates movement in the fluid of seminiferous tubule, and then by virtue of this movement, it sends the spermatozoa out of the seminiferous tubule into the epididymis, which is the first duct of the male reproductive organ. So, myogenic cell. Now guys, the sperm, you have seen a sperm probably. It has a head, it's a cell, right? However, because of its function, has a very long tail, okay? So when they are a spermatid, there is a very huge cytoplasm around them, and they have to shed it. And when they shed it, they become like the conventional spermatozoa. However, the tail of this spermatozoa, which is a protein called flagella, it's a mechanical protein, means that it waves, create movement that allows the sperm to swim, okay? Now, at the time that the sperm matures in the testes, it cannot swim, okay? This tail is unfunctional. Where it matures, it's in epididymis. Later when it goes into epididymis. So this spermatozoa needs to be ejected from the seminiferous tubule, and this myoid cell create movement in the fluid of the lumen of the seminiferous tubule, and that movement pushes the spermatozoa out. Like your children become 18 years old and you push them out of the house by force. I didn't do that, they left by themselves. Okay, so interstitial cell is the fourth type of cell in the seminiferous tubule. Interstitial cells are responsible for production of testosterone, okay? So this is the important slide on the testes and seminiferous tubule, okay? Here is one display, so you can say the epithelial is here, basement membrane, and then these are the 4 steps of the differentiation of the spermatogenic cell. So they give rise to spermatogonia, and then primary spermatocyte, secondary spermatocyte, the spermatid, and finally the sperm. Now those sperm are unable to swim on their own, so they have to be pushed out of the seminiferous tubule by myoid cells. Okay, now, so now we are going to the duct of. So here look at the duct of the seminiferous tubule. Let me see. Okay, so here is the duct. So this is seminiferous tubule, and the spermatozoa are located in the lumen, the ones that have a tail and a head, but not the spermatid stem, are in the lumen, and they pushed into this structure called retitestis. Retitestis. If I have to write it here, this is retitestis. By the way, testis, IS is singular, testis, ES is plural. Another law of plural and singular in Greek language. Okay, retitestis. After retitestis, they enter these efferent ductules, these ones, efferent ductules. After that, they enter into epididymis, which is entire structure. If you open it, it's over 12 meters long. So for a spermatozoa to go from the head of the epididymis to the tail, which is here, takes about 20 days. During these 20 days, the spermatozoa moves and matures and gains the ability to swim on its own, and the flagella becomes active, okay? So the protein that activates the flagella is called dynein. So dynein actually, it's very interesting. So it holds this thing and then moves it. And it's actually real moving protein, okay? So that flagella moves by virtue of those dynein. Now, so after it gets to epididymis, where it goes to ductus deferens, and interestingly, returns to the abdomen. So now we have here mature spermatozoa, and they can swim on their own, and they can tolerate physiological temperature. That's why they go back. So ductus deferens go around the bladder, posterior bladder, and then the end of it swells and become ampulla of ductus deferens. So end of the ductus deferens, it's like, if I draw simply here, swells like that, okay? Then joins the duct of the seminal vesicle, and this new duct that is generated by the, now we need another picture. So another duct that called ejaculatory duct. So let me show you here exactly how it works. Okay, so we are looking at so you can recognize epididymis. Now it enters ductus deferens. Look, ductus deferens enters in the abdominopelvis area and turns around the posterior bladder and then swells. Its end swell. Now you should call it ampulla of ductus deferens, and then joins the duct of the seminal vesicle and the converged duct. Now you should call it ejaculatory duct. Then enters in the middle of the prostate, which you can see here. Now you should call it prostatic urethra. Then it enters at the base of the penis right here. Now you should call it membranous urethra. And then enters the penis itself. You should call it a spongy urethra. So that's the path of the movement of a spermatozoa from epididymis all the way to outside of the body. Now, as I told you, epididymis is a structure in which spermatozoa matures and gains ability to swim, okay? The spermatozoa produce in testes seminiferous tubule and matures in epididymis. And once it gets back to the abdominal pelvis, it can tolerate the physiological temperature. I guess that is it. The ductus deferens, another name, vas deferens, another name for ductus deferens. Now, the glands of the male reproductive organ are three. Seminal vesicle is responsible for production of most of the seminal fluid. And if you don't know, semen and seminal fluid are two different things. Semen is equals seminal fluid plus sperm. Okay? So with the seminal vesicle only produces seminal fluid. Prostate produces 30% of the seminal fluid. Now, the seminal fluid is necessary for a spermatozoa to be able to suspend and be able to swim. And then there is a gland right here, bulbourethral gland. This gland does contribute to the volume of the seminal fluid, but it doesn't produce seminal fluid. It produces a buffer, high pH buffer, known as bicarbonate buffer. Now, if you pay attention in male, the pathway of release of urine from bladder and the sperm are basically in urethra shell. Now, because there is residual urine in the urethra, and urine of the human is highly acidic. And actually, they have done experiment that if you inject 10 ml, only 10 ml of urine into a 5 kilogram rabbit, it kills it right away. So it is toxic. So what you need to do, and a spermatozoa is very fragile, so first bulbourethral gland releases a bicarbonate buffer to neutralize the residual urine in urethra, and then a spermatozoa can pass safely. So bulbourethral gland, if you will be asked, does contribute to volume of the seminal fluid because it's fluid. However, it's not producing seminal fluid. It produces a high pH bicarbonate buffer to neutralize the acidic urine, residual acidic urine in the urethra. So this is basically what we need to know about male. I'm taking it very easy. How many of you know the hormone that makes testes to produce testosterone? It's definitely from pituitary, right? Yes. What is it? Luteinizing hormone, yes. And then spermatogenesis is follicle stimulating hormone, or vice versa. Which one is it? Can somebody check from your endocrine table and get a point? So follicle stimulating hormone causes spermatogenesis, and luteinizing hormone here is responsible for production of the testosterone. Do you remember from endocrine? Confirm? Yes. Where did you confirm it? Where is it? Okay, I believe you. I can't read those handwriting. I'm sorry. Okay, didn't mean to drop it. Okay, now, so guys, so the reproductive organ of male is controlled by pituitary. Is it anterior pituitary or posterior?
Anterior.
Hold on. Anterior pituitary. So posterior is two other one.
Try stream cycle.
We're gonna get to that. Here. I don't read anymore. Okay. So now let's go to this erectile organ of the male, The erectile organ of the male is composed of a very particular structure that engorge with blood. And there are two of them. Corpus cavernosum, it is a pair. So if it is two, you have to call it corpora cavernosa. Write it here, corpora cavernosa. That's a pair of it, two of them, one on each side. Now this is a sagittal view. You can see one of them here, and the other one is on this side. And then there is another structure called corpus spongiosum. Corpus spongiosum. Spongiosum. And corpus spongiosum is only one, and basically surround the spongy urethra. You see it? The spongy urethra is here, and that's corpus spongiosum. So these two organs engorge with blood during erection, and that's the cause of the erection. Of course, it is controlled by autonomic nervous system, and there is a drug called Viagra that basically actually works on the increasing the blood flow specifically to the external genitalia, and basically what happened, this is only good for people who cannot maintain erection. So Viagra inhibit the enzyme that is through cyclic GMP, breaks down cyclic GMP to GMP, and then stops the erection. So Viagra as an inhibitor of an enzyme that extend the erection time, that's it. And then they have made several, today we have dozens of them that they can increase up to 24 hours, means that they slow down the breakdown of cyclic GMP to GMP, so the more cyclic GMP remains in the blood, which is a second messenger, the longer the male can maintain erection. So just one important knowledge that learned from it. Actually, Viagra, when I was a postdoctoral fellow, it was given to patients with cardiovascular insufficiency, and then they did two blind study. So the group got Viagra, and another group got placebo. And people, they were expecting people to come back and say, yeah, we can breathe better, no shortness of breath and things. And then they all came back. There were very inconsistent results about their heart condition, but there was a consistent response about sex life. Their sex life had improved. So they took the label from heart medicine, they put it as a Viagra, and now it's a multi-billion-dollar industry. They are trying to shuffle it in women now, but apparently it doesn't give the same result. In a women, we talk about the erectile organ on next Wednesday. Okay, here is again another view of the ducts of the So one more time, the path of the traveling of the spermatozoa. Now remember, seminiferous does not consider duct of the male reproductive organ. Seminiferous is part of primary sex organ, okay? So we start from epididymis. So the sperm from the seminiferous tubule enter epididymis. Number one. Then enters ductus deferens. From ductus deferens, joins the duct of the seminal vesicle and becomes ejaculatory duct. after ejaculatory duct becomes prosthetic urethra, membranous urethra, and spongy urethra. The reason they call it a spongy urethra, because that corpus spongiosum actually inside of it produces the spongy urethra. Okay, so now do you know that there is a method called vasectomy for people who don't want kids, and the male is ready, is willing to, participate in that goal. So there are two ductus deferens for each testis. So they cut the ductus deferens by a non-invasive surgery, and then they ligate the end of the ductus deferens. So you can imagine that the spermatozoa travels to epididymis from testis and then cannot go to the ejaculatory duct, prosthetic, urethra, and etc. So the male, even though they produce the spermatozoa, they cannot deliver it to the cervix during intercourse or vagina. So what is the problem with this one is that every male produces about 400 million spermatozoa per day. So if you block this, ligate this tube, the spermatozoa enters the epididymis and accumulate, and the epididymis ruptures. And although they tell you sometimes that is reversible, this surgery, but in 50% of people, that ligation creates a scar that cannot be repaired. And second of all, the epididymis rupture as a result of accumulation of all these spermatozoa. Now, what is the problem with this rupturing of the epididymis is the following. Remember, in lymphatic and immune system, we are sending the T cells and B cells to become educated. Do you remember become immunocompetent? Means that they develop receptor for antigen, but not for our cell. So they are not attacking, although we have autoimmune diseases, but let's assume that this is the goal. So they become educated not to recognize self-antigen, but recognize self-protein, but recognize everything that are not supposed to come to our body. The spermatozoa is produced after puberty. So the educated T cell and B cell do not have receptor that knows that these are cells and don't attack them. So the immune system attack the male own spermatozoa, and that causes a severe autoimmune diseases in men. So I think birth control pills and intrauterus devices are safer, although it has to be done by female. Okay? So we talk about this one. Okay, guys. The testis is moved out of the body with a piece of peritoneum and a piece of skin. The skin is called the scrotum, okay? So comes out. But then what if the weather is very cold, okay? And it goes below 3 degrees, like 6 degree cold there, or 10 degree cold there. Like if you walk in Boston at minus 70 degrees. So there are adaptations to protect those spermatogenic cells in cold weather. There are 3 adaptations for cold and hot or physiological temperature for protecting the spermatogenic. One is a skeletal muscle known as cremaster muscle. Cremaster muscle, when it's colder than minus 3 degrees, contract, okay, and pulls the testis closer to abdomen. Pull the testis closer to abdomen, to keep it warmer. What did I do with the male? Oh, there. Okay, so that's cremaster muscle, skeletal muscle. Around the scrotum, there is a smooth muscle. This model doesn't represent any of them, so we have to see what we have. So there is another smooth muscle called dartos muscle. So let's write this: adaptation, cremaster muscle, and dartos muscle. Dartos muscle is a smooth muscle around the scrotum, and when the weather becomes colder than usual and cremaster cannot take care of it, dartos muscle contract and wrinkles the scrotum. Wrinkling of the scrotum prevents heat loss from the testes, and that somehow basically protect the spermatogenic cell. Now, there is also an adaptation. Look at these blood vessels, blue blood vessels. These are called pampiniform plexus. The third adaptation is for cooling down the arterial blood that enters the testis, because arterial blood is 37 degrees, and if it enters the testis, it kills the spermatogenic cell. So this adaptation is called pampiniform plexus. Pampiniform plexus is located under the skin, and basically arterial blood passes underneath, and this pampiniform plexus absorb the heat from the arterial blood, and when arterial blood enters the testis, it's cooler than 37 degrees. So that's another adaptation that helps protect the spermatogenic cell from arterial blood that is 37 degrees. It's known as counter current exchange. Do you know what is counter current exchange? Means that the arterial blood goes to the testes, the pampiniform plexus deliver deoxygenated blood back to the heart, and there is an exchange in between arterial blood and the pampiniform plexus. That's why they call it counter current exchange. You don't need to know it. I never use it in the exam, and it's not on lab practical. I'm just telling you. Okay, so now a few things we need to discuss. I thought we had this model of the pelvis, but apparently none of them. So this one is actually a better model of the female pelvis, more accurate, but that's the only thing we have. So I really don't know if they use that. You're just responsible for that one. But I'm gonna show you this model, lab model that we use all the time in lab practical. And here it is, and I posted it for you on the handout I gave you as a practice. So what do we see here? So you can see the testes undissected, epididymis, pampiniform plexus, corpus cavernosum, because it's one only, corpus spongiosum, the spongy urethra. This is the scrotum. This is the cremaster muscle, the skeletal muscle. Now, the dartos muscle is somewhere here, but it's not represented in this one. It's basically around the scrotum. So that's the cremaster muscle. Now, this is bulbourethral gland. That's prostate. That prostatic urethra. That's the ejaculatory duct. And that's the basically ampulla of ductus deferens, which actually joins the duct of the seminal vesicle and then become ejaculatory duct. Now, this is the bladder. That's the transverse cut of the large intestine. That's called mons pubis, fat in the anterior region of the pelvis. And this one represents, here, in addition to that, you can see the seminal vesicle clearly, prostate clearly, and The rest of them are almost the same. This is just showing the erectile organ in the external genitalia, corpora cavernosa, and corpus spongiosum. They engorge with blood. They are under autonomic nervous system stimulation. So actually, parasympathetic and sympathetic division have now cooperative work. Basically, erection is a result of cooperation of sympathetic and parasympathetic division. Okay, guys, one thing here I want to say very briefly, and I don't include the detail. So spermatogenic cells are basically diploid. I don't know if you know diploid. Diploid in human is 46 chromosomes or 23 pairs. So 2n equals 46. Now, you know that in order to make an embryo or fertilize egg first, you need the sperm to donate its chromosome to the egg, okay? Now, if spermatogenic cells do that, then instead of having a diploid cell, you have 4n cell, which is 92 chromosome. And this is energetically not favorable for every 100 trillion cell of the body to replicate and make them. So therefore, the sperm and egg, the sperm called oogonia and ovum, they go through two phases of cell division and reduction of the DNA. So I don't know if you have learned mitosis and meiosis in biology if you took. If not, don't worry, I'm not telling you. However, 2n means 46 chromosomes. Therefore, this spermatogenic cell cannot donate its chromosome. It goes through a phases of meiotic phases, meiotic 1, meiotic 2, to reduce its chromosome to half. Therefore, the sperm is n equal 23 at the end of this division reduction. So the spermatogonium first divide by mitosis. Mitosis is when you produce two genetically identical cells. Type A, okay, and type B. Type A stays in the seminiferous tubule, like a reserve of the spermatogenic cell, and then type B goes through division reduction, and through meiosis 1. And meiosis 2 produces 4 daughter cells, and each of these have 23 chromosomes, N23. Therefore, when they meet an egg that has gone through the same division reduction, the egg also is N23. They call them haploid. And when they fuse together, now you have a zygote, which is 2n equal 46. And that divide by mitosis during embryonic development to make the 100 trillion cells in your body, in human body. What I want you to learn from this, that the spermatogenesis had two distinct stages. The spermatogenesis and the spermiogenesis. So spermatogenesis go through meiosis and then the spermiogenesis, from which one oogonia becomes 4 complete sperm. That's the point of this slide. Okay? The spermiogenesis refer to those spermatids that have a lot of cytoplasm and they have to shed it to become normal sperm or conventional spermatozoa, and that's the stage of spermiogenesis. basically shedding the cytoplasm and becoming the sperm. And this spermatogenesis occurs in a spermatid phase. So remember, you have the spermatogenic cell or spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid, and a sperm. So here you can see the Sertoli cell or sustentacular cell or sustentocyte that hug all the different stages of differentiation of spermatogonia and protecting them from coming in contact with blood. So they create blood-testis barrier. Now, one more thing here is important, acrosomal reaction. When a spermatozoa arrives to one egg, okay, 400 million spermatozoa arrive to one egg, and I tell you where the egg waits for them. In the reproductive organ of female, the egg waits in ampulla of uterine tube. We're going to learn that next week. Now, so when it reaches the egg, 400 million of them compete for entry of one to the egg. Only one, no more. And this competition is very, very important. You cannot send two spermatozoa and expect egg to fertilize. 400 million create a very high density. They kick each other until one can enter and contribute its chromosome. This is called acrosomal reaction. So in the head of the sperm, there is a vesicle called acrosome, and this acrosome contains hydrolytic enzyme. This hydrolytic enzyme degenerates a protein around oocyte that is called zona pellucida. Zona pellucida. So this acrosomal enzyme can degenerate this protein called zona pellucida, and once they do that, they donate their spermatozoa to the egg. And then egg is called fertilized egg or zygote. Zygote or fertilized egg. Zygote or fertilized egg. So acrosomal reaction is a reaction that the spermatozoa has to go through to be able to donate its chromosome to the egg and fertilize the egg. Okay, here is the hormones that are important for you to know. Not probably, but some of them for lab practical, because remember, Leydig cell produce testosterone and they are in seminiferous group. So spermatogenesis controlled by follicle stimulating hormone. And luteinizing hormone produces stimulate testosterone production, and testosterone itself is very important for spermatogenesis. The inhibin is a hormone produced by sustentocyte that inhibit follicle stimulating hormone and slows down the rate of production of spermatozoa. Okay, this was everything I wanted to say about. Just a brief thing. Prostate is not only a production of the seminal fluid. Prostate has these secrete prostaglandin. I don't know if you know, prostaglandin cause contraction of the cervix and uterus when during intercourse. So it helps the spermatozoa to swim upward. Prostate also produce clotting factors, activate the coagulation, because during intercourse, the spermatozoa is suspended in a fluid. So this clotting, activation of clotting, clots the semen in the cervix or vagina, and then during withdrawal doesn't let the sperm to go backward and maintains it in the cervix or vagina. And then 1 minute later produces fibrinolytic enzyme that basically breaks down the clot and allows the spermatozoa to swim. So prostate is a very, very important, and also it has a pigment that is used in forensic science. It glows green under UV light, shortwave UV light, which has been used for many, many years in forensic science of the rape and etc. So just prostate, this much I want to say. Now that we are done with the male reproductive organ, so as I told you, we have only this model of pelvis. You can take a picture of it because they are the only one that they're going to be using. The one I